Good evening, everyone. Hi, it's 8 o'clock Eastern time. We are here with Fiveable. Welcome. If you've never been to a stream before, we're doing AP Biology on cell size. I'm Kari Parnan, and I'm one of the AP Biology teachers who's also a streamer. And I'm looking forward to just spending some time tonight looking at cell size. Why does it matter? Why do we study it? What do we know about it? All right. Okay. As always, follow Think Fiveable on Twitter and Instagram, and me too, so that you can know when I'm streaming and check that out. Okay, let's get started with tonight's stream. Just a couple things, now that you are in October, happy October 1st, by the way. We've hit October, which is kind of an exciting, scary thing, but you're a decent amount. You should be a decent amount through the AP Biology curriculum, considering the test is in May. So we have October, November, December, January, February, March, April. So you still have 7 months to work with all this information, but we're basically a month and a half in for most schools. So I just have a couple tips. In general, number one, keep up with your assigned readings. I'm sure your teacher or you guys have a textbook of some sort. You should have a textbook, maybe online, maybe paper book. Just keep up with your readings. Read them over, take notes, highlight things you don't remember, highlight things you don't know. And then after your reading, summarize what you've read. So you can even chunk it up and read a small section and try to read it. And then shut your book and try to write it out or talk it out what you just read. And you'll be surprised how much your brain either retains or how much your brain isn't sure about. And then that is kind of the basis for filling in the blanks of what you need. Teaching each other is huge. So like Izzy and Shreya, you guys are in the same class. So definitely lean on each other, teach other kids in the class. So I would pick an objective. If your teacher has a learning objective, like, oh, students should be able to describe the difference between this or that. Have someone read that out loud to you and then try to teach someone else that topic. So teach them verbally or you can write it down, whatever works. But once you really can teach it to somebody, you know it a lot better. I've also talked about learning. So when you're learning horizontally, you're learning stuff that's already been in a textbook, right? So it's horizontal, it's left to right. We're reading what's in the textbook. But then I want you to be thinking vertically, which means don't just think about what's in front of you, think beyond it and ask questions. So like today when we're looking at cell sizes, ask the questions why, how, what would happen if, and just kind of thinking up instead of what's already been answered. Try to think above that. Obviously rewatching streams is great. And then talk it out, draw it out is kind of what I say to a lot of my students. If you can pick a topic and talk it out, just like talk all about it, or draw what that concept is about and what you know about it, it's just going to ingrain it in your brain even better. Okay. All right. So tonight's stream is going to hope to answer the question in a more elevated way. Why are cells so small? And so to get there, we're going to hit surface to volume ratio, adequate exchange of materials, specialized structure of cells, and then a free response and calculation question practice. Okay, so that's what tonight looks like. We have, and the big skill, obviously is performing the mathematical equations, which might seem easy, especially because you're talking about surface area and volume. But when we're talking about something that's in 3D, it does change the equation a little bit. And it's not just a simple square where you're doing length times width for area. So it's good practice to make sure we know that skill mathematically. All right. A review from one of the previous streams about cells. I like to think of all the jobs within a cell that needs to get done. I like to break it up into 4 things. So genetic control, so something to do with DNA, right? Genes are the information that needs to make the protein. Okay. Beyond genetic control, we have manufacturing, distribution, and breakdown. So things are made because of the genetic control. Things are made. Stuff is distributed either out into the cell within the cytoplasm or is distributed out to the cell because it needs to go somewhere else. there's also breakdown that needs to happen. So things need to actually be broken down and taken out. There's energy processing, which is huge, because if a cell doesn't have energy or can't use energy, it's not going to be able to do its jobs like the above, like the manufacturing, the distribution, and breakdown. And the last thing, obviously, you need some sort of support, communication, and movement. And so those four main jobs are where all of our organelles fit in. I like to organize the organelles into those categories. So when we think about genetic control, our mind should think of the nucleus and the ribosomes. Or if we're a prokaryotic cell, I would say DNA and ribosomes. I would not say nucleus, because remember from an earlier stream, we talked about how prokaryotic cells do not have a nucleus. The manufacturing, distribution, and breakdown mostly have to do with your endomembrane system, like your rough and smooth ER, your endoplasmic reticulum, the Golgi, the lysosomes, and the vacuoles. Okay, so by now, hopefully when you see all of those organelles, you should be able to give a rough description of what their job is. Okay, if you can't do that yet, go back and look at our last two streams. Mine was a little bit more baseline understanding of what these things do, and then the most recent stream really went into the details of each of these organelles. So if you are like, what does the Golgi do? I don't know. I would go back and look at either your school's reading or a stream. All right, when we think of energy processing, we should be thinking of the mitochondria and chloroplast. So mitochondria is where cellular respiration happens, and the chloroplast is where photosynthesis is happening. And then cell wall, cell membranes. Some of your teachers might get into cell junctions, but it's basically just the communication between cells is how they're anchored. But your cell walls and your cell membranes are the most important for you to know. Okay, so what are some things, go ahead and type in the chat box, what are some things that a cell needs to do to survive? So what does a cell need to do? Okay, so breathe, some sort of gas exchange, absolutely. What else would a cell need to survive? Replicate, absolutely. It needs proteins, good. Good, needs proteins, needs to make proteins, needs to have proteins. Good, a cell needs organelles, absolutely. What about nutrients? Right, think of like we are made up of millions of cells, right? So we know that our human body needs water. We need to get rid of waste. We need to take in food, right, to get energy. And so on a much smaller scale, because we're made up of organ systems and organs and tissues that are made up of cells, a cell needs everything basically that we need, right? So we need necessary resources, so that whether that be food or energy, like making ATP or just getting a resource, getting glucose, getting rid of waste products, right? Or getting rid of heat or acquiring heat. So even just one little cell needs to be able to get rid of waste. Just like if you think of like eating your whole meal, right? Your digestive system is not only bringing the nutrients needed into the bloodstream, but it's also packaging your waste either into your urine or feces, right? To get rid of it. And so on the smaller scale, your cells have to do the exact same thing. Exchanging chemicals and energy with the environment. So cells need to be able to let things in, let things out, and exchange stuff with what's going on with the environment. Okay, so because it needs to do all these things, the size of the cell matters. So if I go back, if a cell was, for example, huge, right? Its ability to do these three things would be severely hindered, right? So if this is a huge cell, like I imagine a cell that's the size of my body, right? So let's imagine, we know our bodies have millions of cells, but if a cell was the size of me, being able to get in the necessary resources and eliminate waste and exchange chemicals and energy, it would be very difficult and probably waste a lot of energy. Okay, so cell size starts to matter when we're thinking about the compartmentalization of all the stuff that needs to happen for a cell to do what it needs to do. So one of the things I want you to be able to do by the end of today is explain the effect of surface area to volume ratio on the exchange of materials between cells or organisms. So first, we kind of have to break down what is a surface area to volume ratio, and then what happens to it, to that ratio, as cells get big or small. Okay. Bacteria are single-celled organisms, right? So like one organism is just the one bacteria. So the bacteria that makes you get sick, for example, like you get a bacterial sinus infection, right? One little cell multiplies into hundreds and thousands of cells before the infection starts. But if that bacteria was large, its surface area to volume ratio would be different, and therefore it wouldn't be able to be very efficient at its job. Okay, so what I want you to think of, just think of like a simple circular or cuboidal cell. But if size was getting bigger, what happens to surface area and what happens to volume? Okay, so start thinking about that for a second. As size gets bigger, if I'm going from a teeny tiny circular cell to a big, big circular cell, what happens to surface area and what happens to the volume? Okay, so to get there, we need to first think about, well, what is surface area? That's going to be your exposed side. If we're thinking about a cell, surface, meaning it's the surface outside of our cell, okay, area, how much room. Okay, so the exposed side of the cell is your surface area. And then the volume is the occupied space inside of that actual cell. On the AP test, you'll be given this set of equations. So you'll be able to do volume or surface area of 3 different shapes. So either a sphere, rectangular solid, or a right cylinder. So it's fair game, whatever, and you'll have this information, but you'll also have to be able to apply it. It's one thing to be able to do the mathematics and say, okay, I've got it. It's pi r squared times h, and I can get the volume no problem. But then you also have to be able to explain it. So when we talk about the ratio, we're looking at surface area over volume. So whatever you get for surface area, you divide it by volume, and then that's going to be your surface area to volume ratio. And that ratio is what's important. Okay, so I have an example here. So hopefully you have like a scratch sheet of paper near you, because I'm going to test your math right now. I'm going to ask you to look at these 4 shapes, and I want you to tell me, and I'll give you a hint. I would make a chart of some sort, figure out what's the surface area of each shape, what is the volume, and then you're going to tell me what the ratio is. So hopefully you have some sort of piece of paper, scratch piece of paper, make a little chart. We have blocks A, B, C, and D. Okay, you'll be looking at surface area of a cube. So use the equation here for a cube. Get all of their surface areas, get all of their volumes, and then get the surface area to volume ratio. And the numbers here, these are cubes, so every side is the same length. So for A, every side is 40 micrometers. For B, every single side is 30. C, every single side is 20. And D, every single side is 10. So test yourself on this. Actually do this. Make a little chart. See if you can do the math. Yes, so in order to get surface area to volume ratio, I would first get surface area, then get volume, and then do surface area divided by volume, because that's the number we care about. You're not going to see a question that just asks about surface area. It's going to ask for the ratio, but in order to get to the ratio, you have to do surface area and volume for each shape. So when you look at these objects, A, B, C, and D, obviously A is the biggest and D is the smallest. And one who doesn't maybe know much about this ratio or cell biology might say, oh yeah, A has the biggest surface area and volume, and D has the smallest for both as well. So the ratios are probably about the same. The ratio of surface area to volume is probably about the same, right? Because they're cubes and people think like that mathematically. So hopefully you're starting to get it set up. I will give you a look at how I set it up. So I made a little chart the surface area equation was 6, because there's 6 sides, right? Times your, if you go back, it's 6 times s squared. S meaning the sides, like the length of the sides. So 40 squared, or 40 times 40. Volume for something that's rectangular, or sorry, cuboidal, is going to be length times width times height. It is for rectangular too. So once you get your surface area and your volume, you just do surface area divided by volume, and then that'll give you your ratio. So whenever anybody is comfortable, go ahead and type in what the surface area to volume ratio for A is. Awesome. Yes. All right. So 0.15 for A. Good. All right. Absolutely. So I'll just show you the rest of my math there. Absolutely. The ratio for A is 0.15. If you continue the work all the way down, once you get down to D, the ratio is much larger, right? We go from 0.15 to 0.6. That's 4 times larger of a surface area to volume ratio. So it's one of those things where you might, if you try to just do this logically and think like, oh, well, look, A, B, C, and D, you know, we've got a big A and a small block D. Their ratio, you know, their volume and surface area are decreasing or increasing at the same rate, but that's just not true, right? So surface area to volume ratio is the highest in our smallest object. Okay, so I'm going to say that again. Our smaller object, D, has a higher surface area to volume ratio. Because think about it, it still has 6 sides. Every single side matters in terms of surface area. If this was a cell, each of those sides would be a point in the cell membrane where things could get in and out, and it's got not that much stuff on the inside. Okay, so the higher the surface area to volume ratio is actually better. And I know students in the past who have been rock stars at the math, and they can crank out like surface area to volume ratios in 10 seconds, but then they get to a question that's asking them to reason through it and tell me why that matters, and they kind of go deer in headlights, and they're like, I think the smaller ratio makes sense, but it is not, and you've got to talk yourself through it, and we'll go over some more examples in a second. So the higher surface area to volume ratio is best for cell functioning. Okay, so think about this question. Number 1, what happens to the surface area to volume ratio as cubes get smaller? So in this kind of a question, you can look at a data set, and it's kind of like an ACT, SAT style question where you can be like, okay, cube A is the biggest, D is the smallest, my ratio is getting bigger. Good. But that's also something on the AP test, you might not have a data set, and you might just need to know and visualize something that has a large volume and a smaller surface area and what that would mean for a cell. All right, number 2, looking at the data, proportionally, which grows faster, surface area or volume? So now we're kind of thinking backwards. We're saying, okay, from something smaller to bigger, which component is growing faster, surface area or volume? Nice, Sandra. Yes, absolutely, volume. Good job, everybody. Did somebody new just join us? Hold on. Welcome, if we do have someone new. Welcome. We're just going over cell size. We're looking at some data of different cell sizes, and we're realizing that surface area to volume ratio increases as something gets smaller and smaller, which is part of our reasoning of why cells are so small. And then Sandra just answered this question, that proportionally, as you go from something small to big, so D to A, volume is growing at a much faster rate. Okay, so if we go back to an equation of surface area to volume, sorry, let me go back to my equation. Here we go. If we go back to surface area to volume, if our denominator is getting bigger and bigger and bigger, more proportionally, it would make sense that my overall ratio is going to be small when I have a larger object. Ideally, right? We want a large ratio. We want a large ratio. We want more surface area. If you are not in this unit yet, you probably haven't done this lab, but you probably will because it's a really easy lab for teachers to run. I love running it. I've done it a bunch of different ways using kind of like clear Jello. And then we put a pH indicator in there and it like goes from pink outside in. But here, these are actually just beets. So beets are naturally that pink color. And we have 3 different size beets, they're cubes. The beet cubes are the cells, and actually you soak them in bleach, and that's to represent how well are the nutrients able to get in a certain size cell. Okay, so the data is shown there. We could do some numbers and actually measure what surface area actually came through, but just look at it qualitatively. Cell number 3 got a lot of nutrients, right? That's our model. It's not, bleach is obviously not a nutrient, but we're using it as a model to represent things moving into the cell. If you're a cell and need glucose, or if you need anything, potassium, whatever, this is representing its ability to get into the cell and into all the organelles that are in there, because every organelle needs it. Okay, so the bigger cube, look how pink it is still in the middle. It didn't really have diffusion of nutrients that well at all. This would like barely get to the inside of the cell. Okay, that cell would not do very well in terms of surviving if we think back to what a cell needs to do. It needs to get nutrients, it needs to make and use energy, it needs to get rid of wastes. Okay, so number 4, skills check. Answer me 1, 2, or 3. Which beet cube or cell has the highest surface area to volume ratio? Yes, Sandra, you're 3 for 3. Good job. Yeah. So number 3, it is a higher ratio. The higher ratio is better. And I'm probably being repetitive, but I am being so on purpose because students can understand this so well. They get the math. They get the idea of cells are small. Yeah. But then they're not able to reason through, why does this ratio matter? Why can't I just talk about surface area and have tons of surface area? Well, because the volume, right? The surface area is like the plasma membrane. How much membrane is exposed, right? If I'm a cell that's this big, that's how much membrane is exposed. And I have to feed everything that's inside that cell. If my cell is huge, right? I've got this huge membrane. The surface area is bigger, but not as big as how much the volume changed. The more volume means there's more stuff in there, more organelles and more stuff that needs nutrients, and it just wouldn't work out well for the cell. It'd be like this beet right here, where more than half of it, I would say like 80% of it, didn't get any of the nutrients at all, and they were all soaked for the same amount of time. So this is also another piece of evidence. Smaller cells have better and more efficient transportation of nutrients. Oops, I just kind of gave this question away. Hey. Number 5. So fill in the blank. The surface area to volume ratio, the blank efficient a cell is at moving materials into or out of the cell. So A, B, or C. If it's A, the sentence would read, the smaller the ratio, the more efficient a cell is at moving materials in or out. B would be, the larger the ratio, the more efficient a cell is at moving materials. Or C would be, the larger the ratio, the less efficient. Yes, Sandra, you're right. It's B. Absolutely. So we want a large ratio to up the efficiency of the cell. Okay, so cells are tiny because they need to be efficient, and it's due to that large surface area to volume ratio. Excellent. Okay, so thinking about why this is true, again, it's repetitive, but I'm trying to hopefully get it into your head. Sorry, there's a bug in here. You can always walk yourself through it by looking at the equation and say, okay, surface area is on top, volume's on bottom. How could I get an ideal surface area to volume ratio? Sorry, I don't know why that question says 3, but I'm just going to walk you through this one. So for example, if we look at the equation and we want to have a bigger ratio, right? It's going to be either by increasing your numerator, which is surface area. How can I make that a bigger number? How can I increase how much outside is exposed, right? How much of the external surface is exposed? Or how can I decrease the volume, right? Either of those things is going to make your overall ratio a bigger number. Okay, so think about some ways. How does a cell get more surface area, but not necessarily get bigger and bigger and bigger? All right, and some cool ways that cells do this actually. So this is the other part of the AP objective, was being able to talk about specialized cell structure. So not only what is the surface area to volume ratio, why are cells small, but also you should be able to talk about, well, what do cells do to get this number better besides being small, right? So if we look at these 3 cells that I just drew, which one looks more ideal for ideal functioning? So if we're talking about being a really good surface area to volume ratio, think about, we want not just, I think it's pretty obvious that the largest one is not the best, right? It doesn't have much surface area, but it has a ton of volume in the inside. So imagine all this stuff trying to get in and out, and it's got to get to the inside of that thing. Some people are saying top left. Okay, potentially. I tried to draw the top 2 about the same size. So what if I told you that they are the same volume? If the top 2 are the same volume, now which one's better to make my ratio the biggest? We think about what surface area means. It means how much actual surface exposure there is. So the one with all the squiggles actually has more surface area. There's more, yeah, exactly, Sandra. All these squiggles, all these indentations is just more plasma membrane, more entry and exit points for things to get in and out. Okay, so the top 2, I probably should have annotated this a little bit better, showing that they're the exact same volume, but the one on the right with all the squiggles has more surface area, so its overall ratio would be bigger. And Izzy said, yeah, the right one, because it's allowing for more permeability. Yeah, there's more membrane, so the more membrane exit and entrance points, the higher the surface area, absolutely. And so we'll look at some cells that look like that. And there's very particular cells in our human body whose main job is to let things in and out. And it's really, our survival depends on it. And they all have similar structures like that. What about here? I have 3 sets of squares. Each square is a cell. Which group would be best for ideal functioning? I have a 1 by 1 cube, I have a 5 by 5, and then I have a bunch together, but they're also 1 by 1 by 1 by 1. So we have lots of surface area. Imagine each of these squares being individual plasma membranes where things are allowed to come in and out. I should have drawn that as a half, actually, not a 1. So for this one, I was wanting you to just think about the idea of like a grid and how a lot of cells are organized into grids like this. Like your skin cells are organized like this. They're called squamous cells, squamous as in square. And they're like stacked on top of each other like that because they can have great communication, really nice and easy pass-through diffusion of materials that are important, instead of just one cell being by itself is the idea. Okay, keep moving on. So looking at some specialized cell structures, one of the ones that comes to mind in plants are root hairs. So are these cell structures that elongate off to the outside to increase surface area. So obviously if you're a root hair of a plant, your job is to absorb water. And so without those root hair extensions, you don't have a lot of membrane for things to come in. So each little root hair is serving as an extension to that cell to allow for more water. Okay, so more efficient. And we see things like this in plant life all over. Oops, something is up with my slide here. This picture right here are actually the cilia of, my pictures are bad, oh no. Hold on one second. I'm gonna get rid of this root hair picture. Okay, there we go. That's what I was wanting you to see. So the microvilli, this is in your intestines, and your intestines are in charge of absorbing nutrients from the food that your stomach and your saliva have broken down. And so they've broken all this food down into tiny particles, and each of these extensions is an extension of one cell. Okay, so you've got 3 little cells here, you've got 3 purple nuclei, and so these are facing the inside of the small intestine. Those microvilli are increasing surface area, so our cell doesn't have to be very big, but you're going to have a huge ratio, which allows for so much more absorption of nutrients. If somebody is sick, so for example, I actually have celiac disease, and so before I knew I did, my microvilli were all like, they're not nice and filamentous and long. They were like kind of nubbed. And so they had terrible surface area to volume ratio. And so my blood work would come back and it would seem like I was malnourished and anemic. And because basically my intestines, the cells who are supposed to absorb and take in as much nutrients as possible, they were unable to do that because the cells were damaged. And so that's how important those extensions are, because it increases how much more they're able to bring in and how much, if that's their job, is to literally absorb nutrients. When that doesn't happen and that surface area to volume ratio is messed up and not in an ideal number, that's when we're looking at diseases and things like that. All right. Mitochondria. So it has this inner membrane with all these folds. Again, that is to increase surface area. And so that more surface area, once we get into cellular respiration, the more surface area, the more glucose you're breaking down, the more ATP you're making. So if you're making a lot of ATP, then your cell is very, very efficient. Because ATP is like the currency of getting things done in a cell. Last example would be a red blood cell. Red blood cells are this biconcave shape. It's kind of like a donut that's concave in the middle without the hole. That's to increase surface area, which is ideal, because if your job as a red blood cell is to bring oxygen out and take up carbon dioxide, you want to be able to fit as much stuff as possible and have more membrane to let oxygen in and out more freely. If your surface area was terrible, like that's where some red blood cell disorders come from. It's talking about where there's not enough surface area, or it's a sickled shape, the shape of the cell is not right. That can also have an effect of the surface area to volume ratio. Okay, so let's move on to the free response question, and then we'll be done for today. Okay, so the free response question would say, consider the chart and answer parts A and B with supporting details and scientific reasoning. So the chart is giving you sizes in micrometers. You've got a ribosome, bacteria, organelle, and a typical eukaryotic cell. There's two parts to this question. Remember, College Board loves part A, part B. A says, why is it unlikely that a cell would be smaller than 0.1 micrometer? And part B says, why is it unlikely for a cell to be larger than 1,000 micrometers? Okay, so I'm going to give you a minute to just think about the question, reread it. So once you've reread it, hopefully your wheels are spinning and you're like, okay, well, I think I know why it can't be smaller. Okay, but here's where I want to challenge you. Have a supporting detail, at least two supporting details with biological significance for both of these things. Okay, so for example, the first one, I want to give it all away. So I'm not giving you everything. But I would say, you know, cells cannot be smaller than 0.1 micrometers because, A, here's my supporting detail. There'd be no room for organelles, right? I'm looking at the organelle chart and it says 10 to 20 micrometers for just an organelle. So if a cell needs organelles to survive, it can't be smaller than just an average size organelle. So I would cite the chart. I would talk about how all cells do have ribosomes, and so that's the size of the cell they're asking about. Ribosomes are 0.01 to 0.02. Okay, so sure, you could have some ribosomes in there, but you couldn't have any other organelles. And then, even better, I would talk about what an example of an organelle is. So you talk about ribosomes, you can talk about endoplasmic reticulum, you can talk about anything, and talk about what its job is and how a cell would need that. And then also I would cite the table specifically and say the table says this is the size of that, and this is why I don't think it could be smaller. Okay. For part B, why can't it be bigger than 1,000? Okay. Ideally, you would be talking mostly about the surface area to volume ratio here, right? If something was 1,000 micrometers, right? When the chart tells you that eukaryotic cells are max 100 micrometers, that means the surface area to volume would be way too small. And you'd have to explain the ratio. Well, what do you mean? Okay, if it's really, really big, volume increases drastically in number when something gets bigger compared to surface area. So if your volume or your denominator is getting bigger and bigger and bigger, and your surface area is not getting much bigger, it is getting bigger, but not proportionally as big, then your ratio is too small. Talk about why the ratio matters, right? Like in C, I say as the cell size increases, the ratio is going to decrease, right? And we want big, big surface area to volume ratio. Talk about specialized structures found in nature and how important that is for cells to be able to do their job. All right. So I know I kind of went over that quickly. As always, if there's anything more you want to hear or see, let me know. But before we log off tonight, is there any questions? Any questions about cell size? No, I know 44 minutes is a long time to hang on, so I'm really proud of you all for being here. You're doing your part and making sure you're staying on top of your AP Bio. It's huge. All right, no questions for tonight. As always, thanks again for logging in with Think Fiveable. I'm Kari Parnan, so feel free to follow me on Twitter and stay on top of your AP Bio. You guys are doing great. Thanks for coming.